And hey there, microbiology friends. So, let's see. I'll put this in announcements, but just in case some of you are watching, and I hope you're watching, I'm going to post the exam up here in a little bit before I go home today. And it's about, almost 2 o'clock here on Wednesday afternoon. And I can't stand the way Moodle quizzes are set up. I hate them. And the way I want to do the exam is the way I want to do it. I can't fit it into Moodle. Guess what I'm going to do? I'm going to give it to you as an assignment. It will drop whenever I put it on today, and it'll be due Monday. So, it's open book, open note. Go for it. You have no reason other than, to make a perfect grade on it or do really well. Okay? So, do your best. Nope, it's not timed. Yes, use your notes. If you are going to Wikipedia it or Google it, at least put it into your own words a bit. There's a few sort of short answer or micro essays as we think of them, and we'll see where this goes. All right. Hey, now when we last finished up, we were talking about enzymes and specifically how they function to allow chemical reactions to occur at much lower temperatures and much faster. What we're going to do now is get into the overall idea of the breakdown of specifically glucose, and then we'll begin fitting lots of other molecules into this and a lot of other bits and pieces. But there's a pretty seriously fundamental story that I want to talk about that both relates to the biochemistry of bacteria and also the biochemistry of eukaryotes in the way we break down organic molecules. And it's actually a very interesting sort of evolutionary point in that the way organic molecules are broken down in our bodies is very similar to the way organic molecules are broken down in bacteria. I always like to think that, you're sitting around in a bar, sitting next to a bacterium, and you're not going to have a whole lot to talk to them about, but you can say, hey, what about breaking down glucose? Ain't it a pain? Or, I hope you're having a party. You can talk to them about that because they share very similar mechanisms and molecules that we do. So what we're going to get into is initially the breakdown of glucose. And there are a couple of things to think about. First, glucose is a good starting spot. Now, obviously, that's not the only thing that you eat or bacteria eat, but it is what cells often would prefer to eat. And generally, all things considered, if you can convert it to glucose, if you can break it down to glucose, that's what organisms do. So, glucose is a 6-carbon molecule, a 6-carbon sugar. One of the things to be looking for as we're going through this these metabolic pathways is where the carbon's going. Because eventually, our hope is we're going to break the carbons apart and be able to give them off as carbon dioxide. For you and I, we breathe them out. All right. Another thing to think about is the storage of energy and the storage of electrons. And this is where we can begin to think about this metabolism pathway, this metabolic pathway, as a grand opera or a musical theater, is a big story. You got to know your cast of characters. So here are the cast that are going to be involved in this here metabolism play, if you will. We've got glucose. That's the starting point. Now, glucose is going to be broken down, and glucose is going to be broken down into carbon dioxide and water. So that's the endpoints. Now, in order to break glucose down into carbon dioxide and water, we're basically oxidizing it and setting it on fire. And one of the things that we don't have to have, for humans we do, but for bacteria we don't have to have oxygen. But for now we're going to fit that in. And so we're going to go ahead and put here O2. and see how that fits in. So we're going to look at kind of the standard, and then we'll look at all the variations on a theme, how you can do this without oxygen, how you can be anaerobic. Now, that's the beginning, glucose plus oxygen. That's the end, carbon dioxide and water. Now, what comes out of this is energy. Now, energy in living systems is an interesting kind of thing. If we were machines, the energy could be in the form of steam or a boiler or some form of heat. But because of living systems, our energy is coming in the form of chemical bonds. And specifically, energy that we'll be thinking about is primarily in the form of a nucleotide triphosphate that we know, of course, as adenosine triphosphate, or ATP. And just for you super nerds out there, this is a ribose sugar, ATP. This is not the deoxyribose found in DNA. It is the ribose sugar. Irrelevant. The point is, and this is one of the reasons why, that ATP is such an important molecule, is that in order to drive anabolic reactions, now remember we were talking about these are catabolic reactions, but we're doing catabolic reactions to generate energy to build stuff up, to do anabolic type reactions. In order to have an anabolic reaction, we're going to take two molecules. So we have an enzyme that has these two parts. One end is like the big polymer and the other end is a monomer, and we got to slap them together. In order to make covalent bonds, we got to bring the molecules, the atoms, very close together so their electrons can interact. And in order to do that, we're going to have to push these molecules together. There's a little bit of repulsive energy we have to overcome, and so it is a non-thermodynamically spontaneous reaction to build stuff up. And so consequently, we have to put energy into the system. How do we do that? One of the primary ways is by using one of the phosphates from ATP. Now, if you recall back in our early discussions, I hope I mentioned this to you, and I'm pretty sure I did, that whenever I say phosphate, you must always think gigantic stinking negative charge. Phosphate confers a huge negative charge. And so we can have an enzyme that is shaped like this. So a hand, and then another hand. And this hand grabs one molecule, A. This hand grabs the other molecule, B. And we got to slap them together. The way we do that, and the way actually proteins are evolved, is that there'll be a whole stretch of negative charged amino acids over here that are kind of minding their own business. And there'll be one amino acid that typically is a serine or a tyrosine. And this is subject to phosphorylation, having a phosphate added. So when we add this massive negative charge, it is repelling these R groups and causing the hands to slap together. That's why ATP is the energy driving material in our cells. It is the negative charge on the phosphate that interacts with a protein and causes it to change shape. Now, this is a bit of an oversimplification, but it really is a good way to conceptualize, because when you get into anatomy and physiology about muscle contraction, it's all because of the phosphates. The phosphate charge is being added or revealed in a pocket of a protein and causing it to change shape. Now, some proteins will actually have the phosphate covalently linked, and then it can be removed. This is called a phosphorylation reaction or a kinase reaction. Other proteins have a pocket that ATP can fit into, and ATP can break apart, revealing that strong negative charge phosphate, and that can change the shape. But regardless, it is the phosphate that is the driving energy of everything. And bacteria do this, we do this, plants do this. all life on this here planet uses the negative charge of phosphate to drive chemical reactions. And they do it by interacting with proteins and causing them to change shape. I just wanted to throw that in there. That's why ATP is so important. All right. Now, if you recall, we were talking about the fact that breaking down molecules is typically an oxidation reaction. And remember, our mnemonic, oxidation is loss of electrons. Now, if a molecule loses its electrons, it's got to put it somewhere. And we have these little electron dump trucks that are known as electron carriers. And there are three, but we'll talk right now for two primary electron carriers, and there's one really biggie. The electron carriers, they are derivatives of niacin and some of the B vitamins. And they are known, the primary, these are known as, not electron dump trucks, but these are known as electron acceptors. And the primary electron acceptor is a molecule called nicotinamide adenine dinucleotide. Wow. Fortunately, we write this as an abbreviation. We write this as the abbreviation as NAD. That's our primary electron acceptor. There's another one of the B vitamin derivatives based on flavine that is called flavine adenine dinucleotide, also abbreviated as FAD. Now, when we draw them or write them out in this abbreviated form, FAD or NAD, this is the oxidized state. This is the state in which they can pick up an electron. When they are in the reduced state, we write them as, so we can add an electron, and it now is written as NADH. And FAD is written as FADH2. Okay? So that is the two states or two forms of these electron acceptors. They can either be in the oxidized state, in which they can pick up electrons, or they can be in the reduced state, in which they have gained electrons. So, just as a way to, when we're talking about energy, we're talking about ATP. When ATP gives up one of its phosphates, like it's involved in a phosphorylation reaction, it is now written as adenosine diphosphate, ADP, and you have the phosphate coming out. All right? So, here is the point. We've got these molecules, ATP, NAD, and FAD, and they cycle between these two different states. In the case of ATP, they cycle between being the diphosphate and the triphosphate. So we can take ADP, add a phosphate, and convert it back to the triphosphate. The electron carriers, they cycle between these two states, NAD and NADH. You pick up an electron, it becomes NADH. FAD and FADH2. So these molecules, ATP, NAD, and FAD, they're constantly cycling back and forth between the two, their two fates, their two stages, their two phases, their two forms. Let's say that. They're cycling back and forth between those two forms. And when we start thinking about metabolism in cells, there are things going in and things coming out. The only things, though, that are going in are glucose and oxygen, and the only things coming out are carbon dioxide and water. The stuff inside is simply being constantly recycled between these two states, between these two forms. So every cell has got a finite amount of NAD and FAD and a finite amount of ATP, and it's either in the form of NAD or NADH, FAD or FADH2, ATP or ADP. We're just cycling back and forth. You don't add any more of this stuff, and that becomes a critical point because you have to constantly keep recycling it in order to make this system work. All right, so this is our primary cast of characters. We've got glucose, which is the input. That's the carbon going in. We have ATP, and ATP is the phosphorylation doning molecule. ATP can be either in the form of ATP, the triphosphate, or the diphosphate. And then we have our little electron dump trucks, NAD and FAD. When they pick up an electron, NADH, FADH2. Now, let's start working these guys all together. So a couple of things to think about is when we start going through the metabolism is where are these molecules showing up and where are the carbons going and where is ATP being made. So what we're going to do is work through these basic metabolic pathways a bit. And it's a little bit complicated, but I think if you kind of go over it a few times, you can get it. And what we're going to also do is build sort of a cliff notes, real tight little phrases or little things to remember about each part of this pathway that you can easily digest them. All right? So it's like this. In a general sort of sense, and some of these numbers are not exact, depending on the book, it'll be a little bit different, but for our purposes, let's just go with this. Thank you very much. It is this. 1 glucose in the presence of oxygen is going to result in the rephosphorylation of 36 ADPs, which means we say that it's equivalent to generating 36 ATPs. All right? So we want to count, just sort of have our tote board of how many ATPs we're generating in these different cycles. Now, the numbers are not exact for our purposes. We're going to say it's 36, 32, 35, whatever. I don't care. This is the story, and I'm sticking to it. 1 glucose plus oxygen, 36 ATP. So, I mean, in all seriousness, depending on how you want to take it apart, that number can be a little bit different. That's not that it's not this is wrong or the others are incorrect. This is just the way we're going to be presenting it, okay? Thank you. Because I don't want to hear about it. The book said, don't tell me. I don't care. This is my story. All right. 36 ATP. Let's begin. We can break down the breaking down of sugar into 3 separate stages. Some people are lumpers. I am. 3 stages. Some people are splitters, and they do 4 stages. We'll get to that. Stage 1 is called glycolysis. Now, I don't speak no damn Latin, but that is glycolysis, the splitting of sugar. Now, stage 2 is known as Mr. Krebs cycle. It's also known as the citric acid cycle or the tricarboxylic acid cycle, but I like to give Mr. Hans Krebs his due, and I call it the Krebs cycle. The last part of this is the newest part that actually wasn't discovered until like the mid-70s, but there you go. And it is known as the electron transport chain. All right. So these are our three main pathways that we're going to break apart. So we'll go through them, and it'll be a bit detailed, but hopefully at each one we'll finish it and we'll talk about sort of the Cliff Notes condensed version of what's going on. Okay? And so you have a really tight little way of remembering it. Let's begin at the beginning with glycolysis. Glycolysis begins with glucose. And remember, our goal is to phosphorylate 36 adenosine diphosphates to make 36 adenosine triphosphates. The first thing we do is actually kind of backwards, but that's just the way it works. The first thing we're going to do is take 2 ATP and phosphorylate this glucose molecule. And so we generate 2 ADP. And the resulting molecule is something called glucose bisphosphate. But it doesn't hang around very long. It is almost instantly split into 2 3-carbon molecules. We make 2, I don't want to say that, 2 3-carbon molecules. Phosphoglyceraldehyde, for those of you that care, but don't worry about it, you don't need to know it for this class. When you take biochemistry, sure. So, 2 3-carbon molecules. Now, one of the things is that in the textbooks, it often will try and show them either as 2 separate pathways, or they'll sort of like a superimposition. It makes it just that much more confusing, but that's the game. Let's do it this way. We've got now 3 carbons here. And we're going to run this parallel because each one is being operated the same way. The very next step that happens is finally we begin to oxidize this molecule. So now when oxidation occurs, we're losing an electron. One of our electron acceptors has to come in, and that's NAD. NAD comes in, beep, 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 backs up, picks up an electron, and it leaves as NADH. The same thing's happening over here. NAD's coming in and backs away as NADH. The next step, we still have 3 carbons, but now we're going to actually extract some energy from this molecule by taking some of the energy in the bonds and using that to drive the rephosphorylation of an ADP. We actually have 2 ADPs. So we have 2 diphosphates being phosphorylated to form 2 triphosphates. That happens here. Same thing happens on this side. 2 ADPs are being phosphorylated to generate 2 triphosphates. And finally, we're left still with 2 3-carbon molecules. Now, we're not going to get into too many names of the different molecules so much for our purposes, but this is a really important molecule name. So I want you to know this. This is something that is known as pyruvic acid, also known as pyruvate. That's the end result of glycolysis. Congratulations, you've gone through glycolysis. Now, from our tote board, let's do a little tally. What goes in is the 6-carbon glucose. What comes out is 2 3-carbon pyruvates. Now, what also has to be here is 2 NADs. And to get this thing started, we need 2 ATPs. What comes out is 2 NADH and a total of 4 ATPs. 2 from here, 2 from there. So what this means is, in terms of our ATP accumulation, getting towards our goal of 36, we can say this. Glycolysis generates 2 ATP. So, it takes 2 to start it. We get 4 out, a net gain of 2 ATP. Whoopee! We're supposed to get 36. Where's my money? Now, you might say, hey, why don't you put the ADPs in there? Yeah, go ahead, put them in. The point is, if all of your ADPs are in the form of ATP, you don't got to eat. And so, if you're running this, you got plenty of ADP always floating around. So that's why I never really worry about that. In terms of an interesting part of the story, the ADP, it's always there. If it wasn't there, the organism, you, the bacteria, don't need to be doing this. You know, a wonderful biochemical salutation to someone, you know, when you leave them, good luck, good health, would be, may all your adenosines be in the triphosphate form. That means you're healthy, you have lots of food, you are quite well and fed. If all of your ADP or if all your adenine molecules are in the form of the triphosphate, I have to work on that for my logo, but there you go. May all of your nucleotides be in the triphosphate form to your health. All right, we're going to go through one more pathway. The third one, electron transport, is probably worth its own little discussion. So, a couple of things to note is, A, no oxygen is being used here. B, I only got 2 darn ATP and I was promised 36. What the hell? Where are the rest of my ATP, man? So, also, I started out with 6 carbons, still got 6 carbons. You know, it's 2, 3 carbon molecules, but I haven't exhaled a bit. No CO2 is coming out. That's about to change, though. The next step is glycolysis, or from glycolysis is the Krebs cycle. Now, if you recall, we talked about sort of splitting things in terms of the steps, and I like the Krebs cycle, but the next step is this. We're going to take, and this is a note, the 2, 3 carbon molecules are in parallel. They're both going through this. We won't draw the 2 of them. We just know that this one's going in this way, the next one's right behind it or right beside it, doing the same thing. So this is the Krebs cycle. We take the 3 carbon pyruvate and we're going to pop a carbon off as CO2. Finally, we're releasing some carbon. Now, one note, whenever CO2 is being released, that's going to liberate an electron, and so you have to have an electron acceptor right there. That means that you're going to be needing an NAD is there to pick up the electron. So if we have a 3 carbon pyruvate and we've lost a carbon, how many carbons are left? Ain't real high math. 2 carbons. It is an acetate molecule. You remember in organic chemistry discussions that 1 carbon is the methyl group or the formyl group, 2 carbons is the ethyl group or the acetyl group. We've got now the 2 carbon molecule, the acetyl, and it gets complexed up with something called a coenzyme. It's one of these vitamin things. It is a very complicated organic molecule that, why is it stuck on here? It's only on here for a few moments. I don't really have a good reason why. It just is. It's something called coenzyme A. And so this molecule becomes known as acetyl-CoA. All right? It is a 2-carbon molecule. Now, this 2-carbon molecule is going into a rather complicated cycling system. Kind of like it's going to jump on the merry-go-round. And this is a little historical note. Glycolysis was discovered back in the mid-19th century, late 19th century. It's a pretty easy conceptual idea. It is just a straight, long line down. You start with glucose, you break it apart. The next step took a while to figure out. That is the Krebs cycle, because it is not a linear kind of thing. It actually ends up being a cyclic thing. And that took some thinking, and Hans Krebs was the guy that figured this thing out primarily. He actually left Germany after World War I and was working in London. And he brought with him these very elaborate glass tubes, look a little bit like a fancy crack pipe, known as a manometer. And it's a way of measuring very carefully the release of carbon dioxide. And he would add these different components and watch little pieces of liver go through this cycle. And he very carefully figured it all out. And that is now known as the Krebs cycle. It's also known as the citric acid cycle, as we're about to see, also the tricarboxylic acid cycle. So it's also abbreviated as the TCA cycle. I always call it the Krebs cycle, whatever. Now, for this thing to work, we have a 4-carbon acceptor molecule that comes in and picks up the 2-carbon acetyl-CoA. CoA is released, and we now have a 6-carbon molecule, citric acid. In terms of memorizing these guys, if you want to memorize the names, that's fine, but no, for our purposes, we don't need to. We just need to know the flow. Now, the 6-carbon molecule is going to be oxidized and kick out a CO2. How many carbons we got? 5 carbons. The 5 carbon is going to kick out a CO2. How many carbons you got? 4 carbons. Now, this 4-carbon molecule actually undergoes a couple of other rearrangements and eventually ends up at the top of the cycle and is ready to pick back up. So, we are dumping out carbon dioxide here and here. And just as a little point, let's do a quick carbon dioxide tally. We started out with 3 carbons. There's carbon 1, there's carbon 2, and there's carbon 3. And as we said, the second pyruvate is coming in right behind it. And so at this point, we have dumped out 6 carbons from our 6-carbon sugar. Ain't no more carbon sitting in the inside your cells. It's been dumped out. The bacteria are going to allow carbon dioxide to diffuse to the membrane and to the environment, making your beer fizzy. All right. So the next thing that we have to consider, though, is that each time these carbons are being released, here, here, and here, we've got the requirement of an NAD to pick up the electron. NAD picks up the electron. And there are a couple of other things happening that we got to go ahead and fit in here. During one of these reactions up here, we have an electron being dumped out. It's during one of the bond rearrangements, and that is being picked up by FAD to generate FADH2. And we also generate 1 molecule per cycle of a nucleotide triphosphate. Depending on the book that you're looking, it may be written as an ATP. It's actually guanosine triphosphate. It is a GTP. The GDP is phosphorylated. This is another one of those little conventions that nucleotide triphosphates participate in reactions very similarly. Most of the time it's ATP, sometimes it's GTP. Very rarely is it a C or a U or a T, but that's neither here nor there. We throw this in with our ATP count, so let's not be overly persnickety about it, but just to be detailed, there you go. All right, so now if we do our tally, we can say this about the Krebs cycle, is that Mr. Krebs cycle is doing this for us. We got 3 carbons in the form of pyruvate, that is going in, and coming out we got 3 carbon dioxides. Now, if you really want to get picky and count things up, you could. It's not really critical. What we can say is, rather than counting up the NADs that are required, let's just say lots. It's like, 1, 2, 3, more than 3. We got a fair amount of NAD being required here. So let's just go ahead and say lots of NAD is got to go in, and 1 FAD, and what comes out then is lots of NADH, 1 FADH2, and 1 nucleotide triphosphate. All right? So if we go to our tote board over here, we can say that Krebs cycle is generating a grand total of 2 nucleotide triphosphates, 1 per cycle. Now let's just groove on this for a second. I was promised 36 ATPs per glucose. I have dumped all of my carbon dioxide, or all my carbons out as carbon dioxide. There ain't nothing else there to chew on. And now I've got 2 ATPs from glycolysis and 2 nucleotide triphosphates for our tote board purposes. It's an ATP. 2 ATPs from the Krebs cycle. That's 4 ATP. I was promised 36. Where the hell are the rest of them? I want my ATPs. We're going to have to wait until next week to figure that out. But that actually is an important thing. We have not generated all the ATP that we was owed. Oh no. We are, in fact, missing 32 ATP are still left out to be extracted. What we do have is lots of now reduced electron acceptors, and we have to recycle them. So just as a little preview, the last part of this system is known as, it actually comes under a couple of different names. It is called the electron transport chain. It's also called oxidative phosphorylation, which is, as we'll begin breaking this thing down, an absolutely stupid term. It doesn't make a lick of sense, but it could be called the Great Phosphorylating Kablooey, would probably be a better term for it, but they didn't ask me. So, the electron transport chain is doing a couple of critical things. And when we get into it, this is really sort of the Cliff Notes way to remember what the electron transport chain is doing. Rather than trying to draw it out, what is it doing? Number one, it is recycling. Yes, that should be one word. NADH back to NAD, so we can go back up and pick more electrons up. It's recycling FADH2 back to FAD. And as a little side note, in systems that are using oxygen, and not all will, but this is the place to start, we can say this: O2 is the final electron acceptor. Now, as we get into a bit more of the details of different metabolic pathways in bacteria, we'll see that O2 doesn't have to be the final electron acceptor in all systems. Sometimes there are some bacteria that use sulfur, but we use oxygen, and aerobic bacteria use oxygen. And so, for now, let's just throw this in sort of a generalized way, and then we'll fit it into the specialized ways. O2 is the final electron acceptor, and it's like this. We've got O2, and we're going to add electrons. That is going to generate a molecule of water, and since there's only one oxygen in water, we're left with half of a diatomic oxygen, or an O1, which this is not written that way. This is known as the oxygen radical. It is the oxygen that's in hydrogen peroxide. It's very, very bleaching, very oxidative. So the biochemists would have us write it as this: 1/2 of an O2 molecule. Now, the next electron coming in will slam into that one and generate another molecule of water. So if you want to balance things, you could just say 2 molecules of oxygen and 1 molecule of oxygen and 2 electrons, and you make 2 molecules of water. But regardless, this is how we're going to leave it, all right? So that's what the electron transport chain is doing. It recycles NADH back to NAD, FADH2 back to FAD. Oxygen is the final electron acceptor. And it then is going to generate ATP using a gradient of protons, which I'm not going to write up because we're going to get into that our next time. So, the exam will be laying out soon, and good luck to us all. I'll check in with you. I'm actually not going to be around here Friday, so we won't do another lecture until Monday, but I'm around and online. Good luck. Peace.